Hi everyone, and welcome to another recorded lecture of BIO101 online. Today we will discuss Chapter 8, which is on cellular respiration. This is actually the last chapter in the first unit of the textbook. So let's start out with a quick question. So take a second to pause here and try your best to answer this rapid response question. So of course you should have read the book already. That's the best way to learn. And you should know that the answer is C. Plants absolutely carry out cellular respiration, as do all living things, because all living things require ATP, and respiration generates ATP. How much ATP do you think is made per second in one of your cells? So all of your cells require ATP to function, but think about how many molecules of ATP could be made per cell in 1 second. 10 million. So your cell, each one of your cells can make 10 million ATP molecules per second. Again, impossible to imagine. You have about 37 trillion cells in your body. So that means every single second, you're making about this number of ATPs. That's 37 with 19 zeros. Or 18 zeros. 3.7 times 10 to the 20th power of ATP molecules being made every single second in your body. So clearly, cellular respiration must be a remarkable process that works very efficiently. Right? And of course, we rely on photosynthesis and respiration to live. So these processes have been fine-tuned over billions of years. So here we see a picture of an ocelot that's eating a lizard of sorts, and it is breathing in the oxygen from the atmosphere, and it is consuming the nutrients from the lizard. And together, these nutrients, which include glucose and the oxygen from the air, enter cells. And in the cytoplasm and mitochondria, the process of cellular respiration will break down that glucose and those nutrients using oxygen in the atmosphere to make ATP. That is the goal. Making ATP. As a result, right, the waste products are CO2 and water. Looking at this picture, we see this plant here is also doing cellular respiration. But this plant doesn't need to eat a lizard to get its glucose and nutrients, right? What does the plant have to do? Right? It just needs water, sunlight, and CO2. But still, if we zoom in on this plant still has mitochondria that are using oxygen to break down glucose into carbon dioxide and water and making ATP as a result. So it's very important to understand that all organisms do cellular respiration. Only plants and some algae and bacteria do photosynthesis. So all cells need chemical energy. The chemical energy all cells require is specifically ATP. But we can't just eat ATP, right? We can't go to the store and just have an ATP milkshake. We have to have food. And molecules in food store chemical energy in their bonds. So if you were to go get a sandwich, that bread is made up of a lot of starch, right, which can then be broken down into glucose. And every single bond that you see here, either within each glucose subunit or between the glucose subunits, contains energy. So we can harvest, right, all the energy contained in these bonds and transfer it to ATP so our cells can use that energy. And when you eat that sandwich and you eat that bread, right, those nutrients in the bread were essentially produced by autotrophs using photosynthesis. Right? So where did the starch in your bread come from? Well, it came from the wheat plants. Right? The wheat is actually a fruit of the wheat plant. And of course, that the wheat plant was using it to store energy, but we're going to use it to make bread. So the wheat plant did photosynthesis to take the energy from the sun and to store it into starch. We're going to eat the starch and then convert that starch chemical energy into ATP. Right? In cell respiration, the energy from nutrients is used to make ATP. And cell respiration is exergonic because ATP powers the cell, right? And cellular respiration requires oxygen to be most efficient, and it produces carbon dioxide. So we'll talk about a way that cell respiration you can get away with not having oxygen, but to make the most ATP, you need oxygen. We therefore say that it's an aerobic process. Aerobic means oxygen is required. Cell respiration is also referred to as, aerobic cell respiration is called the Krebs cycle, or the citric acid cycle. I would take a second to just make note cards or some kind of a chart so you don't get confused between the Calvin cycle, which is part of photosynthesis, and the Krebs cycle / citric acid cycle. It's a lot of redundant names, so it's really important to make sure you get the vocab right so you don't get confused. So let's use an example of a pizza, right? Pizza is an energy source. This pizza is made up of carbs, fats, and proteins, right? There's fats in the meat and the cheese. There's carbs in the crust and the sauce. So when we digest this pizza, right, degradative reactions, right, catabolism, will break down our molecules, those pizza molecules, into subunits. So the proteins will be broken down into amino acids, the carbs will be broken down into glucose, the fats will be broken down into fatty acids and glycerol. These digesting processes tend to be exergonic. They release energy. So what we're going to do is use the energy released from these molecules and store it in ATP. So all of these building blocks of life are basically going to be inputs into cell respiration. We can use amino acids and glucose for energy. Right? So ATP is generated from the energy in these food molecules. So specifically, fats break down into glycerol and fatty acids, and parts of those molecules can enter this cell respiration. Amino acids get broken down into carbon chains and amino groups. The amino groups get removed and they get converted into urea by the liver, but the carbon part enters cell respiration and can be used to make ATP. But what we're going to focus on today mostly is how carbohydrates are broken down into glucose and how glucose enters the cell respiration pathway. So we're going to talk today specifically about how glucose gets broken down in cell respiration to make ATP. But just know that when you have proteins and fats, those can also enter cell respiration and be used to make ATP as well. Okay, so the inputs to cell respiration are glucose and oxygen, right? The outputs are ATP, CO2, and water. You must know these formulas, right? And it's pretty easy to remember if you remember photosynthesis because it's the reverse of photosynthesis, right? Photosynthesis, we said light energy plus water plus CO2 gives us oxygen and glucose. Now we're doing the opposite. We're saying glucose and oxygen give us energy in the form of ATP, CO2, and water. This occurs in the mitochondria of eukaryotes and the cytoplasm. Obviously, bacteria don't have mitochondria, so they do all of respiration in their cytoplasm. But we have cell respiration occurring partially in our cytoplasms and partially in our mitochondria. So is cell respiration endergonic or exergonic? right? It's exergonic. It's releasing energy. We're going from a high energy reactant starting material to a very low energy product, CO2 and water. Right? This is exergonic. This will be a negative delta G reaction. It is oxidation. So this leads me into the next slide, because cellular respiration, just like photosynthesis, is a redox reaction. Right? Something is getting oxidized, something is getting reduced. Specifically, glucose is being oxidized into CO2. Remember what oxidation means. It means the loss of electrons, which means the loss of hydrogens. It's breaking down. So this glucose is losing it's losing hydrogens, it's losing electrons, and it's breaking down into carbon dioxide gas. Oxygen accepts electrons and protons and becomes reduced into water. So oxygen gets reduced into water, glucose gets oxidized into CO2. Recall that photosynthesis is the opposite, right? And again, you must know what gets oxidized and reduced in each respiration and photosynthesis. So you can pause here and try to answer this question. So chloroplasts, which are in photosynthesis, convert blank energy into blank energy. Right? The answer is B, solar energy into chemical energy. Cellular respiration is a blank process. The answer is B, exergonic. Right? We're releasing energy. So what I want you to focus on, perhaps more than anything in this chapter, is how photosynthesis and cellular respiration are related. The two reactions are so fundamental to life that every student studying biology just must be at least familiar with the two reactions and how they're related. So I don't want you to focus as much on the textbook and memorizing the numbers of all the products that are made and all the names of the enzymes. I want you to know mostly how photosynthesis and respiration are related to each other and what the inputs, outputs, locations, and functions are of each step of respiration. So let's start talking about how photosynthesis and respiration are related. In cell respiration, we're making ATP from glucose and other food molecules. Right? And the ATP is made in the mitochondria of plant and animal cells. And where does the glucose come from? The glucose comes from photosynthesis in the case of autotrophs. Right? Autotrophs make glucose from sunlight in photosynthesis. Heterotrophs need to eat and digest food to absorb glucose and nutrients. So let's look at the two equations and see how the two reactions are connected. It turns out, like I said, the reactants in photosynthesis are the same as the products of cellular respiration. So in photosynthesis, CO2 and H2O go in, and out we get sugars and O2. In cellular respiration, the reactants are the same as the products of photosynthesis. Right? Sugars and O2 go in to make CO2, H2O, and energy. So what these organelles, the chloroplasts and the mitochondria, are doing, they're just converting one form of energy to another. Right? These are like little conversion factories. They're just converting one form of energy to another. The chloroplasts are converting solar energy into chemical energy of sugar. The mitochondria converts the chemical energy of sugar into the chemical energy of ATP. And of course, heat is lost anytime you transfer energy from one form to another. So make sure you're familiar with these equations. And everything definitely does exist in a delicate balance, as Mufasa says in The Lion King. Right, so this plant is using carbon dioxide and energy from the sun to make sugar molecules in photosynthesis. So this plant is sitting out of the sun, this tree is sitting in the sun and making its own food. As a result of making its own food, In photosynthesis, it releases oxygen. This animal over here can breathe in the oxygen and will consume other producers like this grass, and together will be able to make ATP using cell respiration. Right? So the animal takes in oxygen and breaks down the sugar molecules contained in the grass. In respiration, the animal releases carbon dioxide, which again is one of the inputs of photosynthesis, which is required for plants to make more sugars. So this is the delicate balance that, life depends on. And this is why if you cut down trees, you're going to have an effect on other living things, not just that forest. Right? Everything depends on each other. And chloroplasts and mitochondria themselves are related because both organelles are involved in reduction and oxidation. So take a look at this diagram again and make sure you understand what this is depicting, how the inputs go into the chloroplast in photosynthesis, and how these outputs in plants would go into respiration in the mitochondria. Right? Carbon dioxide is being reduced in photosynthesis, and carbohydrates are being oxidized in cellular respiration. Remember, energy does not cycle between the two organelles. It flows from the sun first and through each step of photosynthesis and cellular respiration. And eventually, the energy becomes unusable heat when the ATP is used up by the cell. So recall the second law of thermodynamics. Right, so every time we convert energy from the sun to sugar to ATP, energy is being lost as heat. And that's increasing the entropy in the universe. So this is a table that I would like to be in your notes. This compares both photosynthesis and respiration. And it talks about whether food is being produced or consumed, is energy being stored or released, is light required, and then for H2O, CO2, and oxygen, is it being required or released? You should also know where each process takes place, whether it's endergonic or exergonic, anabolic or catabolic. So again, this is a good way to organize the information. So pause here and try to see where oxygen is labeled in this image. The answer is A. Right, so this is a good figure that you could expect this on the midterm and being asked to label A, B, C, and D in this image. Where is CO2 labeled? So the answer is C, is CO2. Water is labeled D. And what is B? The answer is C, glucose. So I just want to also take a point, make a point of that respiration is not the same as breathing. So a lot of times we say like the respiratory pathway, right, is the pathway of the lungs. But breathing refers to gas exchange in the lungs when you inhale and exhale. That's breathing. Respiration is using oxygen to convert glucose into ATP. So respiration occurs in cells. And of course, all cells require energy. So respiration is occurring in almost all of your cells. But some cells that require a lot of energy do respiration more and have a lot more mitochondria. So muscle cells, for example, have a lot more mitochondria than a skin cell would. So remember that respiration is gas exchange in a way because you're using O2 and you're releasing CO2, but it's not the same as breathing. So let's take a second to watch an overview of cell respiration. Again, I only want you to know the information presented in the slides themselves, along with the tables I give you in the learning map. So if there's some information in the animations that is not mentioned elsewhere in the slides, you don't have to know it. but sometimes I want to give you the bigger picture in an animation so you could have that information as well. I want you to know as much as possible without overwhelming you. So let's take a second to watch this overview.
In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide.
Great. So I mentioned before that cellular respiration is a redox pathway. A lot of reduction and oxidation is going to happen. So in order for reduction and oxidation to occur, we need enzymes. So respiration requires many enzymes to work in a pathway. And we spoke about enzymes in a previous chapter, and we said that oftentimes enzymes require other molecules to allow them to function. And for respiration, B vitamins are required as coenzymes for these respiration enzymes to function. So the reason why we need B vitamins is to make ATP. Without riboflavin and niacin, you might recognize some of these ingredients on the back of a cereal box, right, that are found in wheat, and are sometimes added in as a supplement. So oftentimes B vitamins are added just because we do need these. These are essential coenzymes for our respiration pathway to work. But there are 2 main coenzymes that I want you to know. One is called NAD+, and the other is FAD. NAD+ is a coenzyme that's made from vitamin B3. So that's why we need vitamin B3, for example, because NAD is made from that. And NAD plus, I should say, is like the uncharged battery, right? This has no electrons. That's the oxidized form. But when it accepts electrons, it gets reduced into NADH, and that's our charged battery. Each NAD plus molecule can be used over and over again. Note, NADP plus is used in photosynthesis. So you can remember the P for photosynthesis. So NAD is in respiration, NADP is in photosynthesis. Another coenzyme is FAD, which becomes FADH2 when it accepts electrons. So what we're going to see in respiration, very similar to the light reaction in photosynthesis, is we're making energy storage molecules. We're trying to charge molecules up so we can then use them to make ATP. So what we're doing is we're going to try to make as much NADH and FADH2 as possible from our glucose, so we can then use those energy molecules to make ATP. So I want to break down the 4 phases of respiration, and this is the basic version, so you should know this. Glycolysis is the very first part. Glyco refers to glucose, lysis means breaking down. Glycolysis is the first part of respiration where glucose is broken down into 2 molecules of pyruvate. So glycolysis is the breakdown of glucose into 2 molecules of pyruvate, and this occurs in the cytoplasm. Pyruvate is a 3-carbon molecule that will feed into the next phase. So some ATP is formed in glycolysis, but we're not getting much from glycolysis. The main purpose of glycolysis is to break down glucose into pyruvate in the cytoplasm. This process is entirely anaerobic, meaning oxygen is not required. So some organisms can do glycolysis to get their small amount of ATP without any oxygen. So this is not efficient without oxygen, but it's still better than nothing for some organisms with no oxygen exposure. The next phase is called the prep reaction. And what happens here is that pyruvate is oxidized by a special enzyme before it could enter the next phase. So it's just preparing pyruvate for the next step. Energy is stored in NADH in this step as well, but no ATP is produced. So this is a very quick step. We're just prepping pyruvate for the next phase. In the prep reactions, CO2 is released as well. The main phase of cellular respiration, the one that requires oxygen, is called the citric acid cycle. This is the one you should definitely be familiar with. This is also known as the Krebs cycle. The citric acid cycle occurs in the matrix of the mitochondrion and produces lots of NADH and FADH2. So what the citric acid cycle is going to do, it's going to oxidize molecules in order to reduce NADP plus and FAD into these high energy electron carriers. Right? So it's a series of reactions. It's a cycle. And as we oxidize molecules, we're going to release carbon as CO2 and transfer that energy into these energy carrying molecules. Since glucose has to be broken down into two pyruvates, the cycle has to turn twice per glucose molecule. And we only get 2 immediate ATPs per glucose from the citric acid cycle. So it's not very efficient. All we're really getting from the citric acid cycle is NADH and FADH2. Those are the main products of the citric acid cycle. The last phase of respiration is the electron transport chain, which should ring a bell because we spoke about such a chain in the chloroplasts in photosynthesis. But the electron transport chain in respiration is a series of carrier proteins on the cristae of the mitochondria. So those are the inner membranes, like the foldings, right? Remember how it folds over and over again? Those are the cristae of the mitochondria. So these proteins on the cristae of the mitochondria will be able to extract the energy from NADH and FADH2, passing those electrons from high energy to low energy. That will finally, those electrons will be passed on to oxygen. This will allow water to be formed as a final product. So the electrons get passed from one protein to the next. Finally, oxygen accepts those electrons and it becomes water. We could then use all of the electrons' energy to pump protons across the membrane and then use that potential energy to make ATP by chemiosmosis. So just like in photosynthesis, how we pumped protons out of the thylakoid to try to make this concentration gradient and to use that concentration gradient to make ATP, we're doing the same thing. By passing the electrons from NADH and FADH2 from protein to protein, we could use that energy to pump protons inside the intermembrane space of the mitochondria, and we're going to use that concentration gradient to make ATP by chemiosmosis. So this is the overview of every step we're going to talk about. This is a way to organize some of the information. So for each of the 4 phases, where in the cell does it happen? What's the main goal? What are the major inputs and outputs? And you don't have to know anything in this. I would like you to know only what I wrote already. You don't have to add anything into this column. And then you can tell me, is oxygen required, yes or no? So again, limit your studying to mostly what's on this table and on these slides. The book goes into a lot more detail. And of course, there's a lot more detail that we could go into beyond this textbook. So the first step we said was glycolysis. So glucose cannot just enter the mitochondria on its own. First, we have to break it down into two pyruvates. This happens in the cytoplasm. So we start off with a glucose. We end off with two 3-carbon pyruvate molecules, 2 ATPs, and 2 NADHs. So we do get a little bit of energy from glycolysis, but that's not what we're doing glycolysis for. We're doing glycolysis to break down glucose into pyruvate. In order to start glycolysis, you actually need to input 2 ATPs. At the end, we end up getting 4 ATPs because special enzymes add a phosphate to ADP. So substrate level phosphorylation means an enzyme can take a phosphate from a product and add it to ADP to make it ATP. This is different from chemiosmosis, which was how ATP synthase made ATP. Right? Do you remember the spinning of that enzyme, ATP synthase, allowed a phosphate to be added to ADP to make ATP. But here we're making ATP in a different way. It's a less efficient way. We're making it by substrate level phosphorylation, which means an enzyme adds a phosphate to ADP to make it ATP. Since we needed to put in 2 ATPs and we get 4 ATPs, that's how we output a net of 2 ATPs. Right? We need to put in 2, we got 4. So in the end, we have a net of 2 ATPs and 2 NADHs out of glycolysis. So that's what you have to know for glycolysis. You do not have to know this diagram in the book, but this shows you how 2 ATPs are used to get started. The 6-carbon molecule gets broken down into 2 3-carbon molecules. NADH gets made, ATP gets made, and then there's a little rearrangement, and pyruvate is formed. So we have ATPs, 2 NADHs, and 2 pyruvates. This is a little more detail, but you can watch this video on your own, which is included in the Connect ebook. But this just shows you a breakdown of how we get those pyruvates. So, step 2, the prep reactions. So pyruvate cannot enter the citric acid cycle yet. We first want to get it from the cytoplasm into the mitochondria and convert it into acetyl coenzyme A. Acetyl coenzyme A is what can enter the Krebs cycle, the citric acid cycle. So what we have to do is take the pyruvate from the cytoplasm, transport it into the mitochondrial matrix, that's where this occurs. And to pyruvate as input, it's transferred into the mitochondrial matrix, and it is broken down into acetyl coenzyme A and CO2. Pyruvate had 3 carbons. And notice this is CO2 right here. So when you clip off the CO2, you end off with a 2-carbon product. So acetyl coenzyme A has 2 carbons. You also get NADH, but no ATP is made at all. So what we did in the prep reaction was convert pyruvate to acetyl-CoA, and now we're inside the mitochondria. So far, we gained 2 ATPs, 4 NADHs, and 2 acetyl-CoA from 1 glucose. Nothing exciting happened so far, right? We didn't really get much. But now we're in the mitochondria for the Krebs cycle, also known as the citric acid cycle. For every glucose molecule, meaning for every 2 pyruvates, energy will be extracted from those molecules, right? Those molecules will be oxidized, and as a result, we can charge or we can reduce NAD plus and FAD into their higher energy molecules like FADH2 or NADH, And we can then use those molecules to make lots of ATP in the last step. We also make a little bit of ATP in the Krebs cycle by substrate level phosphorylation, meaning an enzyme adds a phosphate to ADP to make some ATP. All of this occurs in the matrix, and it starts, right, well, this is taking you from the glycolysis. This is pyruvate. This was the prep reaction. We removed the CO2, we made an NADH, and we started off with acetyl coenzyme A. This is the first starting product. This is the input into the Krebs cycle. The reason why the Krebs cycle is called the citric acid cycle is because the first step is to add this 2-carbon acetyl coenzyme A to a 4-carbon molecule to make citric acid. Citric acid is a 6-carbon molecule. So it's citric acid that's going to be oxidized step by step in a cycle, and energy will be released stepwise, little by little. So citric acid will be oxidized, and CO2 will be released at each step. Right? So I want you to understand the basics and the concept of this, right? We build a 6-carbon molecule that will then feed into this cycle. CO2 will be released, right? We're breaking it down. And as we're breaking down citric acid, we're going to store that energy into energy-carrying molecules like NADH and FADH2. It's important that the release or the breakdown of this is stepwise. Right? We want to make sure we don't lose too much energy as heat. So if we converted all the glucose into ATP all at once, we would lose a lot of that energy as heat. So by doing things little by little, very gradually, we're minimizing any heat loss. At the end, we get only 2 ATPs, which is not that great, but we get 6 NADHs, 2 FADH2s, and 4 CO2s. And the important part are these energy-carrying molecules, because this is what will go into the next and final phase to oxidative phosphorylation in the electron transport chain. Oxidative phosphorylation is like chemiosmosis. It's the way that we're going to use ATP synthase to make lots of ATP. Right? So this is the overview in the book. Here's this citric acid molecule, and step by step, there's oxidation reactions. CO2 is released, NADH is made. CO2 is released, FADH2 is made, and so is ATP. Right? Here's some more NADH being made. So what we're doing is just making energy-carrying molecules and releasing CO2 in the citric acid cycle. So this is a good diagram to keep track of, right? We started with glycolysis in the cytoplasm. We put in our pyruvate. We did a prep reaction, the citric acid cycle, and now we're finally ready for the last and final part, which is the electron transport chain and chemiosmosis. So again, I recommend watching this video on your own, just to get acquainted with the actual steps and what's going on in this citric acid cycle. It's good to know what's actually going on inside of your cells. So far, we haven't made many ATPs. We only made 2 from the Krebs cycle and 2 from glycolysis. Not very efficient. But how do we produce lots of ATP at once? The electron transport chain. The electron transport chain will allow us to make lots of ATP by chemiosmosis and oxidative phosphorylation. So NADH and FADH2 will carry electrons and the protons received from the Krebs cycle to the electron transport chain in the inner membrane of the mitochondria. So remember those cristae, those folds? And we said all the ATP making enzymes are on those membranes. So that's why it has to fold to maximize surface area. This is what we're talking about. The electron transport chain is found on those inner membranes that are folded over and over again to maximize surface area. So we want to get the most ATP production in a tiny little mitochondrion, So what's happening here? We start off with NADH, which is, again, it has electrons on it. And what it's going to do, it's going to it's going to transfer its electrons to electron carrier proteins on the membrane. And these electrons will pass along from one protein to the next along this chain in the membrane. Specifically, cytochrome proteins are shown. They're little carrier proteins that diffuse through the membrane. They require iron to work, cytochrome proteins. They could carry the electrons from one protein complex to the next. And as these electrons are transferred from one protein to the next, the energy from the electrons is used to pump protons, H+, out of the matrix and into the intermembrane space. Remember that this Krebs cycle, the citric acid cycle, happens in the matrix. So what we're doing now is we're using all the NADH and the FADH2 in the matrix. We're transferring those electrons to the proteins along this inner membrane, and we'll use those electrons to pump tons of protons in here. We're putting tons of protons, which are H pluses, in this dark red area. That's the purpose of the electron transport chain. We're taking the electrons that were carried by NADH and FADH2. We're having them being transported along proteins in this membrane, using those electrons for energy. And then we can actively transport H+ into the intermembrane space. This creates a proton gradient. Right now that we have tons of H pluses here, we could use this gradient to drive ATP synthesis by chemiosmosis. So remember, as H pluses, which really accumulate in the intermembrane space, as H pluses accumulate, they can then go from high concentration to low concentration. Right? We can use that potential energy to drive the kinetic energy of this ATP synthase molecule, which can make ATP by what's called oxidative phosphorylation. Importantly, oxygen is the last electron acceptor. If oxygen is not present, the electrons cannot complete their chain, and their ATP will not be able to be made. Oxygen is absolutely required to receive the last electron and to be made into water. If that does not occur, if oxygen isn't present, ATP synthase cannot move, which is why if you're deprived of oxygen, you die. Right? You cannot make ATP if you don't have any oxygen because there's nothing to accept the electrons and the electron transport chain. So you won't be able to make ATP. So we said ATP allows protons to go back from the intermembrane space into the matrix, down the concentration gradient, to make ATP by ATP synthase using chemiosmosis. And we can get up to 34 ATPs per glucose with this enzyme. So compare that to making just 4 ATPs this whole time. Now we can make up to 34 ATPs just in this last and final phase. So again, this is showing you more details than you need to know. It's showing you the specific names of the enzymes. I would love for you to know that, but I'm not going to test you on it. So here you can see step by step how electrons are being transferred from NADH from one protein to the next. This is a cytochrome protein. And every time electrons are passed to another protein complex, protons will be able to be pumped into the intermembrane space. Here we go again. This will be able to carry electrons over here, which will then release more protons. Finally, oxygen can receive these final electrons and turn into water. And then we're going to use, so again, this happens all the time. This is a constant change. Remember how many ATPs we're making? 10 million per second. All the H pluses can then go into this ATP synthase, and make lots of ATP. Right, and it's called chemiosmosis because this chemical H plus is going from high concentration to low concentration, and ATP synthase works by oxidative phosphorylation. So to summarize, we said glucose in the cytoplasm got broken down into 2 pyruvates. We got 2 ATPs. We then had to prep it to make 2 acetyl coenzyme A. We got some NADH from that as well. The acetyl coenzyme A fed into the citric acid cycle, which oxidized these molecules to reduce NADH, or to make lots of NADH and lots of FADH2. As a result of those breaking down, we had a lot of CO2 released into the atmosphere and a little bit of ATP. Together, all of the NADH and FADH2s feed into the electron transport chain, which are the inner mitochondrial membrane that form those cristae. And together we get up to 34 ATPs. So together, from glycolysis, the prep reaction, and the electron transport chain, we can make up to 38 ATP molecules per glucose. This is about a 40% conversion efficiency. So we're extracting about 40% of the energy in glucose and converting that into ATP. And that might sound inefficient, but compare that to a car. So a car only uses about 20% of the energy contained in gasoline to physically move. So it's very inefficient when you think about it. So our mitochondria are twice as efficient as an automobile engine. So let's watch this video to review.
The energy within a glucose molecule is stored in its chemical bonds. Cellular respiration is a 4-stage process which releases this energy and forms ATP. These reactions occur both in the cytoplasm and mitochondria of the cell. The first of these reactions is called glycolysis, and it occurs in the cytoplasm of the cell. A series of chemical reactions breaks down glucose to pyruvate, in the process generating a net of 2 ATP molecules. In addition, a small amount of NADH is generated, which will be used later in the electron transport chain. As the pyruvate molecules move into the mitochondria, they enter the preparatory reactions. These reactions rearrange the chemical bonds of the pyruvate, forming acetyl-CoA. In the process, some NADH is released. Following the preparatory reactions, the acetyl-CoA molecules enter the citric acid cycle, also referred to as the Krebs cycle. Here, the remaining chemical bonds are broken, generating 2 molecules of ATP, carbon dioxide, and several molecules of NADH and FADH2. The final set of reactions are collectively called the electron transport chain. The inputs to the electron transport chain are the NADH and FADH2 molecules from glycolysis, the preparatory reactions, and the citric acid cycle. This stage of cellular respiration uses chemiosmosis to produce the majority of the ATP needed by the cell. It is here that oxygen is used as an electron acceptor, producing small amounts of water. For each glucose entering these pathways, between 36 and 38 ATP molecules are produced.
So this is a very good animation to watch over and over and over again until you feel like all these concepts click. So here is a filled-in version of the chart. I would highly recommend not looking at this and actually testing yourself with the blank chart first. But you should definitely know everything as far as location and main goals. And as far as inputs and outputs, focus on the bold things, the things in bold. And then I said you don't have to really know the key, any other enzymes that are not listed here. So now let's quickly talk about anaerobic respiration. So when oxygen is not available, cells can make ATP using glycolysis, which makes a little bit of ATP, followed by fermentation. So if you don't have oxygen, it's not a huge problem. And fermentation is an anaerobic process that produces a limited amount of ATP when oxygen is not available. So fermentation produces a lot less ATP than aerobic cellular respiration. In yeast cells and some fungi, they generate ethyl alcohol and CO2 from fermentation. So this is the reaction of fermentation in yeast. Glucose gets broken down into carbon dioxide, ethanol, and ATP. And you can think about this in terms of beer. So beer is made from malt, wheat. You're going to learn a lot about, or actually you've already hopefully read all about beer in our relevancy module. But the energy contained in sugar in the beer comes from wheat at one point, or the barley, the malt. So that sugar gets broken down by microorganisms, yeast, right? Consume the sugar from the carbohydrates, like maltose, right? Malt is beer. So maltose is a disaccharide that yeast can break down. And as a result, they make ethanol, which is alcohol. That's why beer is alcoholic. And they make CO2, carbon dioxide. That's why beer is carbonated. So when you think about fermentation, think about beer, right? It's carbonated, it's alcoholic, and it was made using maltose, malted sugar. And we get only 2 ATPs. So it's really not that efficient. It's only about 2% as efficient for the total complete breakdown of glucose. Right, so compare 2 ATPs by fermentation compared to up to 38 ATPs by aerobic respiration. Lactic acid fermentation is carried out by certain bacteria and fungi, and instead of producing ethanol, we produce lactic acid. You might be familiar with some like the muscle burn that happens after strenuous exercise. And that's because your muscle cells have used up all of their oxygen, so they can't do aerobic respiration anymore, right? They can't have oxygen as a final electron acceptor in aerobic respiration, so muscle cells can resort to lactic acid fermentation to break down glucose to make a little bit of ATP, while you try to recatch your breath. But what happens is lactic acid accumulates. And lactic acid can be poisonous if it accumulates. So it actually causes a burn. And the burn you feel is the burn of lactic acid, which is being produced by anaerobic respiration when your muscle cells don't have adequate oxygen exposure. So the advantages of fermentation is you get a quick burst of ATP energy. Like I said, when your muscles are working vigorously, lactic acid fermentation can provide bursts of ATP. But that lactic acid or the ethyl alcohol is both toxic to the cells. And the lactic acid changes the pH of your cells, which causes that muscle soreness. It's something called oxygen debt. So what you have to do after you consume too much oxygen is you have to recover. You have to take deep inhalations. Right? Like after you sprint, you have to stop to catch your breath and recover the oxygen debt. So here's some rapid response questions. I'm just going to show you each of them. Once you see them, please pause, take a second or a minute even to think about the best answer. This is good practice for the midterm, which will be multiple choice and short answer. So where does glycolysis take place? The answer is A. Which of the following is not a product of glycolysis? The answer is C, right? Glucose is a reactant. Which of the following can carry electrons? The answer is C. Which of the following is an input to the Krebs cycle in the mitochondria? The answer is A, acetyl coenzyme A. In the electron transport chain, protons are pumped from the blank into the intermembrane space of the mitochondria? The answer is B, from the matrix into the intermembrane space. In which phase is ATP produced most? The answer is the electron transport chain. Right, that was where we got the most energy. So let's conclude by going back to the energy organelles. And as you've studied photosynthesis and respiration, I hope you made a lot of connections between the two processes. Right, there's a lot of connections to be made. And in all organelles, structure determines function. And there are numerous examples of how the chloroplasts and mitochondria have adapted their structure to perform their specific function very efficiently. Right, so this has occurred over millions of years, right? If not billions of years, you can argue. So the membranes themselves are really remarkable, right? The membrane of a chloroplast forms these thylakoid membranes, right? Remember, that's what has the photosystems on them to absorb the energy from the sun. So these chloroplasts maximize their ability to absorb solar energy by folding their membranes into thylakoids, just like the mitochondria's inner membrane form those cristae, which maximize ATP production. Also recall that both of these organelles were thought to be independently existing on their own, right? That's called endosymbiosis. So these organelles have their own DNA and divide on their own as well. The electron transport chain is found in both of these organelles on the membranes. So this has been, again, like a very efficient process. The electron transport chain in chloroplasts is located on the thylakoid membranes. And in photosynthesis, electrons are passed to the electron transport chain that were first energized by the sun. Right? So chlorophyll has electrons that are energized by the sun. Those electrons get passed along the electron transport chain to make ATP. In the mitochondria, the electrons were energized by glucose, essentially. Right? Glucose was oxidized. That energy was transferred to NADH. And NADH and FADH2 transfer those electrons along the cristae to move H pluses against their concentration gradient. So lots of ATP can be produced by chemiosmosis. So we see the same exact thing, right? Making the electrochemical gradient in both organelles. So try to, again, always reinforce these connections. Another connection is that just like the matrix of the mitochondria and the stroma, right? Those are the liquidy spaces around the organelles. That's where all the enzymes are. So in the chloroplast, the stroma has all the Calvin cycle enzymes, and in the mitochondria, the matrix has all the Krebs cycle enzymes. So again, here's another similarity. And a great way to end this unit is to kind of synthesize all the information we learned since chapter 1. We said all living things require energy to function. And energy flows from the sun through chloroplasts to carbohydrates, then to the mitochondria, to ATP molecules. So this flow of energy is crucial to all of life on Earth. And of course, this flow of energy is what maintains the biological organization at all levels, from atoms to the biosphere. Right? Remember that atoms and molecules like to disperse. Things don't like to be organized. We need a constant input of energy in order to maintain organization, in order to maintain life. And all this energy ultimately comes from the sun. And at each step of the energy transformation, we lose some energy as heat. Right? So all the energy from the sun is eventually lost as heat, and all life depends on a constant source of solar energy for that reason. Right? We can't just harvest all the energy from the sun and keep on using it. It eventually all gets dissipated into the universe as unusable heat. So we need a constant source of solar energy from the sun in order to live. So while energy flows, right, in a constant renewal of solar energy, chemicals cycle. Right, so chloroplasts produce oxygen and carbohydrates, which are used by mitochondria to generate ATP, which is required for life. So this is like a system that's a cycle that feed into each other. Right, photosynthesis and respiration are directly related. Right, and chloroplasts and mitochondria allow the energy to flow through organisms and allow these chemicals, the carbohydrates and CO2 specifically, to cycle. So in conclusion, we see that all living things depend on a constant flow of energy and a constant cycling of chemicals. So this is the end of the unit. I hope you were able to gain a new appreciation of what makes things alive, how they use energy, how they're organized, and how they use enzymes and other macromolecules to function. And we're going to continue talking a lot about cell function in the next unit. We're going to focus mostly on DNA, genetics, and biotechnology. So this is all the information you need for the midterm. And I will be posting a review sheet to help you review on everything that's going to be covered. So you can complete the lecture review assignment, which is on both photosynthesis and respiration. There's no quiz on Chapter 8, but Chapter 8 will be covered on the midterm.